Okay, so for this lecture, this covers chapter 17, which is the cytoskeleton. So we'll be talking about the different filaments that make up the cytoskeleton. So we're going to look at actin filaments, intermediate filaments, and microtubules. So this figure is from another textbook, but it takes one of the figures from our textbook and basically overlays them. So just keep that in mind, and we'll look at the figure from your textbook in the next slide. But this is a nice figure because it shows these three levels of filaments that are found that make up the cytoskeleton. And so in that red pink color, you can see the actin. So you'll notice that the actin is going to be found in highest concentration near the surface, near the plasma membrane. So it is found throughout the cell, but again, the highest concentration is going to be in closest to the plasma membrane. And that the actin filaments are also responsible for any kind of projections. So here we can see these microvilli in these epithelial cells, and I would imagine these are lining the gut. And so these microvilli, these projections, are because of actin filament. So actin filaments are going to be the smallest filaments because they're between 5 and 9 nanometers in diameter. Besides microvilli, we would also see actin responsible for those projections like pseudopodium that we would see in amoeba. Okay. And then we have our intermediate filaments. So these are in between in size of actin and microtubules. So the way to remember, if I was like to ask you, like, which is the smallest and which is the largest, what one falls in between. Intermediate filaments were given the name because they actually are in between size of the actin and the microtubules. So they're about 10 nanometers in diameter. So these are in the figure are the blue threads, and so they actually allow for this mechanical strength of the cell. So keep in mind that cells are dynamic, they're making up tissues, and those tissues are constantly moving. So again, if this is in the intestinal tract, when you eat food, material is going to be moving through that, and the cells have to be able to kind of flex with that and stretch, and so allowing that lumen on that apical side to open up more. And so intermediate filaments allow for that kind of flexibility, so that there's a kind of a stretching of the cell without the cell actually breaking apart or shearing. Okay. For the microtubules, these are shown in the green. So these are going to be about 25 nanometers in diameter. So these are really important for cell division and cellular transport. So for cell division, we need to have two cells, one cell dividing into two cells, and so making sure that organelles are separated properly, and that you can get that pinching in of the plasma membrane. So microtubules kind of orchestrate and allow for all that to happen. For cellular transport, microtubules are going to be kind of the highway that allow for vesicles to move along them. Okay. So this is the figure that's from your textbook, and what it does is it just separates the different filaments into their own column, opposed to having them overlaid into one cell. So here we can see, starting from the left to going to right, we have the intermediate filaments making up the blue. You'll notice that this is throughout the cytoplasm of the cell, but we also see that it is isolated in the nucleus, so it's going to make up kind of the framework, the shape of the nucleus, known as the nuclear laminar. Okay, which is important for giving the nucleus its shape, but also it creates kind of a mesh so that it can control proteins coming in and such. Okay. Microtubules being in the middle, so you can see these are isolated more towards the center of the cell. You can see that lighter green, which is the centrosome, and that's going to be our microtubular organizing center. So we'll see that microtubule development comes out of the centrosome, and microtubules are going to grow in a fashion away from the centrosome. So the centrosome is kind of like home base, and so they we'll talk about how they grow and the capping that goes on with them. And then lastly, we've got the actin filaments, and so again, you'll notice that they're going to be concentrated towards the outside of the cell, or those projections. That there is some in the cytoplasm, but it's more of a interweaving, which we'll see in a little bit. So looking at intermediate filaments, the panel on the left, panel A, is immunofluorescence, where they've used antibodies to stain keratin in the green. So keratin would be the intermediate filaments, and there's associations with gap junctions or cellular junction proteins, such as the desmosomes. And so they can they're anchored on those. And so that's what's shown in the blue, are those gap junctions, desmosomes. On panel B, we can see this is a drawing trying to show what we're seeing here on panel A. So do keep in mind that one of the things that the textbook has done, depending upon your edition, you might have different colors. So I actually have the 5th edition of the textbook, and the colors for panel A are different. So they're inverted. So it's keratin is blue and the junctions are green. So just be aware, don't memorize the color, know the pattern. Okay, so kind of that spider web appearance with the lack of the staining, where the nucleus is, would be where our keratin is, our intermediate filaments. Okay, the reality is it's the center of the nucleus that's not staining. There's going to be intermediate filaments making up that nuclear lambda. Okay. And again, if you look closely on panel A, you can see these points where you can see there's connections between the two cells. So if you look kind of at the cell that's kind of in the middle, and then towards the top of it, to the right, and also to the left at the tip of it, you can see these points, and that's where desmosome is. So desmosomes are kind of like Velcro. They anchor two neighboring cells together so that it helps in stabilizing and giving strength, right? Because again, these cells are going to have forces on them. And so these desmosomes with the intermediate filaments allow them to be structurally stronger. So when we look at the intermediate filaments, they're going to form multiple lateral contacts. And so what that means is if we look at these proteins as they're coming together, they're going to have connection points among them. And that's what's going to give them their strength so that they can deal with those forces that would otherwise rip them apart. And so they end up having a very rope-like property, right? So if you were to look at a rope or a thread, it's not one single fiber, it's multiple fibers that are actually spun together. So, if you know someone who lives on a farm with sheep and everything, and they shear the sheep, they take the wool, and one of the things is you have to start getting the fibers in the same way. So you use like a comb to do that. But then through using a spinning wheel, you're actually going to spin those fibers so they're actually intertwined together, so that thread has a stronger properties than they would individually. Okay, and this kind of staggering assembly of these of these proteins, and then cross-linking the proteins together allows it to be very strong, but at the same time being very flexible. So just like a rope, it's not going to be, it's not like a PVC pipe that's rigid and can't flex and everything. So a rope has a lot of flexibility because of these multiple points that are coming together. So as I mentioned, there's this mechanical stress flexing that's going to be happening of cells, and so intermediate filaments allow for cells that are going to be constantly exposed to stress, like the ones in your GI tract, to be able to deal with that. So what this figure is just showing you is how we have this monomer that's going to come together to form the dimer, and you might have remember from Unit 1 talking about coiled coil. So these dimers are going to flex together. So now they're going to come together with another coil to form a trimer, and so, but that's going to be staggered. So that's what we were talking about before, is like there's going to be like an overlap in them. So then your trimers are going to come together. So we have 2 trimers, and we have the staggering and these cross linking points. So some examples of intermediate filaments, keratin filaments are found in the cytoplasm. So those would be found in epithelial cells. Pimentin's another fibrous protein that makes up the intermediate filaments. So these are commonly found in connective tissue or muscles or glial cells in the nervous system. And then we can also have neural filaments. Filaments are specific to nerve cells. Okay, when we look at the nucleus, this is where we can have fibrin, and so that's the nuclear laminin that makes up that. So that would be found in animal cells. So if we have a mutation to the keratin genes, what ends up happening is that we don't get attachment of that epithelial layer. So we can actually get this shearing off of the epithelial layer. And so here we can see A would be a normal tissue, and then B is the mutant. And so we can see how that connective tissue that would be holding those cells together isn't able to do that. And so on C, we just have a kind of cartoon of what's happening. And again, we end up having this shearing effect so that there's separation between the cells. Okay. And then lastly, in that lower panel, it just shows you a C. elegans, which has a mutation to its keratin gene, and where there's the arrows, there's separation between the membrane and the cuticle, which is the outer layer of the C. elegans worm, which is a nematode. It's a roundworm. So the specialized intermediate filament that's making up that nuclear lambda in the nuclear envelope, you can see here in the drawing right near that nuclear pore, we have some of those fibrin filaments near there. And if you went back to Unit 2, when we were talking about transporter proteins, when they're being transported from the cytosol into the nucleus, they have to go through the nuclear pore. And in there's some of those fibrin filaments that create almost that basket that allow to regulate which proteins are going to come in, because larger proteins wouldn't be able to come in because the basket would catch it from being able to go into the nucleus. Okay. But as here, you can see that there's this underlying throughout the nuclear envelope layer of these filaments. And again, it allows for there to be a framework, a mesh in order for the nuclear envelope to keep the structure of the nucleus. So an important thing to keep in mind is that intermediate filaments are not going to just be interacting with themselves. They're also going to interact with the other cytoskeleton components, other filaments. So here we can see in the blue we have our intermediate filament, but we also have in the red the microtubules, and you'll see the green. The green is plectin. Plectin is actually going to kind of act almost as like a anchoring tape to tape those intermediate filaments and those microtubules together as they intertwine with each other, right? So it's important that we have these intermediate filaments interacting with the microtubules, because we'll have these vesicles and potentially organelles moving along those microtubules, and we want to make sure that they're anchored, right? So that if the cell flexes or changes its shape in response to something, that the microtubules aren't just floating out there, in the middle of the cell. They also have anchoring, and so plectin is the anchoring protein that keeps those interactions stabilized. The last piece in this figure is a yellow, and the yellow is just showing the immunogold staining that's specific to plexin. So they used an antibody that had a piece of gold on it, and that antibody recognizes plectin, so that you're able to, it's like immunofluorescence. You know that's the protein you're looking at because the antibody is specific to it, and the gold allows us to be able to see that using electron microscopy. So with electron microscopy, you don't have the lasers to see different colors. So you have to use the gold or heavy metals in order to see that. So if you have a scanning electron microscope facility and you're going to do immunogold staining, you have to buy gold labeled antibodies. And most of these universities, they see that on the invoice and they're like, "You bought gold? Why did you buy gold?" So now looking at microtubules, microtubules are going to be made up of tubulin. So think microtubulin, but it's not microtubules. So here we can see the network that the microtubules are going to make up. So keep in mind that the figure we saw before, the first couple slides, it looked like there was like 2 or 3 microtubules in the whole entire cell, but it's more of a network. So that was, some of these drawings are simplified so that you can see all the pieces to it. So this is a better visual for panel A, because this is immunofluorescent staining for tubulin, and you can see all those thread-like structures, and that's going to make up this network that's going to allow for structural support, but also transport through the cell, right? So along all those little threads, we could have vesicles moving through the cell. So keep in mind in Unit 2, when we were talking about vesicle transport of proteins using vesicles, and that all the transport between the different organelles. And the reality is there's really another piece to those figures, and that would be the microtubules and the motor proteins that are involved. Okay, so microtubules are important for many different cells. So non-dividing cells are going to have the microtubules that again will help with movement of vesicles and organelles through the cell, so transporting of material, so cargo to different locations. But then for a dividing cell, it's important because the centrosome, that microtubule center, they're going to separate, and they then become the poles of the mitotic spindle. So if you remember back to like an intro bio class when you were talking about mitosis, we need to be able to take chromosomes, we need to be able to separate them out, so that there are a set of chromosomes into each cell, and we need to divide the cell. So we have, cytoplasm for each cell, we have ribosomes for each cell, organelles for each cell, and so the microtubules are really important in order to do that, and the centrosome allows for that organization to occur. Then we also have specialized applications for microtubules. So that would be for cilia. So if a cell is ciliated, this is where those microvilli would actually beat or wave. If it has cilia, then it will actually have a basal body. The basal body acts as like a motor in order to coordinate the beating, the flexing of the microtubule back and forth. Okay, and then a really specific microtubule and kind of an extension of the cilia would be a flagella. Okay, and so we also have that basal body on a flagella, and it's going to coordinate that movement. Okay, so we'll talk a little bit more about that. So next we're going to talk about microtubules. So we're going to talk about the assembly of microtubules. So the microtubule is made up of a heterodimer of tubulin. So there is two subunits, and so there's an alpha subunit and a beta subunit. So you can see here in panel A, we have the alpha subunit on the bottom and the beta tubulin subunit up above that. And then you'll notice that each one of these tubulin subunits is binding a GTP molecule, which is shown in the red. Okay, so the alpha subunit is actually going to bind to its GTP tighter than the beta subunit is going to bind to its GTP. Because of that, this causes kind of a shift in this heterodimer so that you end up having a rotation occur. So as you can see in panel C, because of this rotation, we end up creating almost like a, circular staircase, if you will, of these heterodimers. And then there's a lumen in the middle. Okay. So the other important property with microtubules is that they have a polarity. So they have a plus end and a minus end. So as you can see in the panel B, we have the minus end marked at the bottom and the plus end up on the top. And if we carry this over to panel C, you can see how on the plus end, this is where we're going to be adding these heterodimers. So the minus end can add them, but they don't add as quickly. So if we look at this in this figure here, we have cilia that's been isolated, and so there's tubulin available for the cilia to grow. So you can see how we have our plus end. So our plus end is where we're going to have faster growth, and that would be because the K on, or the rate of new subunits adding on, is going to be faster than the K off, or the rate in which subunits are coming off. Now in the minus end, it's going to be slower, or a shrinking that's going to actually happen, and this is because the K on is happening at a slower rate than the K off. Okay, so you can kind of think of if you have a high rate of subunits adding on, you're going to be growing and forming, extending the microtubule, right? But if you have a higher rate of K off, then that's where you would get shrinking or decrease growth. And again, in the plus end is where we're going to have the growth. And this ability to control the growth relates to whether the GTP has been undergone hydrolysis. Remember, hydrolysis is where we're removing a water molecule, and for the GTP, it would be converted to GDP. And so you can see in this figure how at our bottom we might have a mix of it, but when we start having more of our subunits in the D form where we have GDP, it actually is going to slow down the rate in which subunits can be added on. Where on our plus end, hydrolysis doesn't occur as quickly, and so we're going to have more of those subunits having the GTP, so in the T form, so we can more quickly add in additional subunits onto this polymer, right? And so the same process is also happening for actin, but with ATP and ADP. So as the microtubules have grown, and if the microtubule needs to be stabilized, what can happen is a GTP cap is going to be put on. So that cap would prevent for there to be like shrinking a loss of those tubulins. Okay, and again, GTP cap is generally going to be put onto your positive end. Okay. So another process that can happen is what's known as treadmilling, and we'll talk more about this later on. But treadmilling is where you're going to get an addition of a monomer on the positive end at the same rate in which you're losing a monomer at the negative end. So that rate of adding and subtracting is going to be the same. So there's actually no growth that happens. So it's kind of like, you know, you're on a treadmill and you're putting one foot in front, but at the same time your foot then moves back. So you're not gaining any distance. You're not walking down the hall. You're staying in the same spot. And so treadmilling is the fact that again, if you're adding monomers at the same rate that you're losing them, then there's not going to be any lengthening of the microtubule.
Microtubules continually grow from the centrosome added to a cell extract.
Okay, so the other thing is if you end up losing the GTP cap, then that's going to start, that's going to signal for a shrinking. So here we can see we have growth occurring, and then there's something that happens that causes the loss of the GTP cap, and you can start to see how there's a decrease. So then, in order to rescue this, you have a GTP cap put back on, right? So towards the left of that GTP cap, we can see how we have some of those GDP subunits. And then this would then allow for regrowth to occur. So looking at microtubules, the centrosome is going to be the microtubular organization center, or MTOC, that most animal cells have. This allows for microtubules to grow from this. So tubulin would kind of be located in that location, and then we can have growth out. So we can see on our cell here on the left how we have the positive, the plus end, moving away from the centrosome. So then once the microtubules have reached the length that they need to be, they can be stabilized by putting capping proteins on. So here we can see how these microtubule capping proteins are placed onto the microtubules so they no longer grow. And then we have our unstable microtubules, if you can panel D, so they don't have the cap, and so they could continue to grow. So this is one way in which cilia become stable, so that they can maintain a certain length. There are there's some drugs that we can use in the lab that can affect microtubular permeabilization. Taxol is one that's probably most commonly known. It's also a cancer drug. And so what it does is it will stabilize the microtubules so that they're more or less capped so that we can't get this, they don't become as dynamic, so the cell can't respond. So it kind of like freezes, halts the cell. So again, these drugs are also used in cancer treatment because cancer cells need to be able to rearrange their microtubules so that they can undergo cell division. And so this prevents them from being able to do that. So in this figure, what we're looking at on the left in panel B is a liver epithelial cell, normal. So we have the microtubules labeled. And then panel C is after Taxol treatment. So you can see how the microtubule pattern is very different. So it's not going to be able to, the cell's not going to be able to move material around on it. So as I mentioned before, the function of microtubules is to act as a transport, a highway, if you will, to move vesicles and organelles around in the cell. And so the movement can either be away from the cell body or towards the cell body, right? So you've made a microtubule, and then along that tubule we can move material, cargo. And so if it's in an outward fashion, so what we see, the red, so we can see these red vesicles that are moving towards the positive end. So in a nerve cell, this could be neurotransmitter, and so we need to get that neurotransmitter out to the axon terminal so it can go to the synaptic cleft, right? So this is going to be outward transport or entrograde. Now if we were going to be bringing something into the cell body from the positive end to the negative end of a microtubule, this would be known as retrograde transport. So again, it's inward, and that's what we're seeing for those blue vesicles. Transport is moving towards the cell body. So here we can see in panel A we have our fibroblast, so we have our centrosome, and then coming out of that are microtubules, so our positive ends are towards our plasma membrane, and we can see that we have the movement of vesicles with kinesin moving out towards those positive ends, and then the vesicles that are using dynein are going to be moving in towards the centrosome, those blue vesicles. Okay. And so depending upon what direction the vesicle has to go, it would affect which motor protein is aiding in its movement. Now, the neuron that we looked at before, we were just looking at the axon, and so we could see that the plus end was all moving away from this centrosome, away from the cell body. You'll notice our dendrite that we do have microtubules that are moving in both directions, so that's very unique. So just be aware that some cell types have unique microtubule orientations and arrangements. So what we're looking at here is an axon, and you'll notice there's a white frame, and that white frame is in a fixed location, and we have mitochondria that are lining up along with the microfilaments, and you'll notice that there are these two vesicles in the frame on the left that are in that white circle. But as we go from left to right, this is looking at different time points. It is 400 milliseconds between each frame, each picture. And so you can see how those vesicles within that white circle have moved when you compare the first frame with the second frame. By the third frame, they're starting to move outside of that white circle. By the fourth and fifth frame, they're no longer in that white circle. And so what they're doing is those vesicles are moving along a microfilament to move to a different location. Okay, so mitochondria is also going to have to do that. So other organelles would also use the microtubules to move along the axon. So what is driving all this movement? It's going to be motor proteins. So these motor proteins have these globular heads, and the globular heads actually walk along the microtubule. In class, I'll share with you a video. I'll also post the link of the video. But there's this animation that was done showing these vesicles being moved along a microtubule. Is pretty cool. But they had these like little globular heads, and they look like little feet walking along the microfilament. So two of these motor proteins that are important for microtubules are diane and kinesin. They're going to move in different directions, and this allows for vesicles to move in either towards the positive end using kinesin or towards the minus end using diane. Okay, and as the panel B shows, it's almost like one globular head moves forward, and then the next, as that one gets put down, the next globular head moves forward, and that tail would be holding on to the vesicle so that the vesicle or the organelle would be moving along that microtubule. But that motor protein, diane or kinesin, is what's anchoring them to the microtubule. So it's kind of like someone walking on a balance beam, and I don't know, holding something above their head. And so each step, even each movement with that globular head is driven by ATP. Okay. So there are going to be adapter proteins that are going to allow for those connections between the motor proteins, diane and kinesin, and the cargo. And the cargo would be the vesicle or the individual protein that's being moved, or the organelle that's going to be moved. So in this figure here, we can see how these microfilaments are going to be used as a scaffolding, a highway for organelles to be found along, because if organelles need to be moved, they would be able to be moved along these microfilaments. So you can see how on this figure, the blue is going to be the endoplasmic reticulum. So kinesins are going to be moving that endoplasmic reticulum out away from the centrosome, away from the nucleus. So it's actually moving it away from the centrosome, but the nucleus is going to be located near the centrosome, where diane is going to be pulling and keeping the Golgi, which is in the yellow, towards the nucleus, towards the centrosome. So the centrosome is going to be located again near the nucleus. And those microtubules are going to be shown in the green. So it'd be nice if they did like a little overlay of these. But again, you can see how in panel C, the upper panel has the Golgi in yellow. You can see the nucleus is kind of blocking. It's almost like the moon going in front of the sun during an eclipse. And then in the lower panel, you can see all of the microtubules that are present in those locations. And again, it's the dianin that's going to be keeping that Golgi anchored near the nucleus. Now with cilia and so next we're going to talk about cilia and flagella. So cilia and flagella are made up of stabilized microtubules, as we can see on panel A, the left panel. Those hair-like structures are cilia, and inside of each of those hair-like structures, we're going to have a microtubule, so made up of tubulin subunits. And then there's dianin is going to be the motor protein working with the basal body to allow that movement to happen. So the basal body is going to act like the centrosome, so it's going to organize and be the base that the microtubule is going to be formed out of. In the dianin, and we'll look more at this, but the dianin is going to be the motor protein that's going to allow for the flexing and that movement back and forth of the cilia and flagella, which is known as that movement is known as the power stroke. So the arrangement of these microtubules is going to be what's known as a 9+2. So you can see how in panel A and also in panel B, you can see these microfilaments that are microtubules that are formed, and so there's 9 that pair together on the outside, and then 2 additional ones in the middle. So there ends up 9 times 2 is 18, plus 2 in the middle, 20. So there's 20 microtubule filaments that come together to end up creating the cilia and the flagella. Okay, so flagella structurally are very similar to cilia. It's just elongated so that the microtubules are going to be longer, and there's going to be there's more. You're going to end up finding mitochondria at the base of a flagella, and mitochondria spread throughout near the location of cilia, because you need ATP in order for dianin to be able to move these microfilaments. So if you look in panel B, you can see that we have, in the red, we have the dianin, and so these are, you can kind of think of this as like a ratchet. It's going to be grabbing, and so that's going to allow for this kind of turning and flexing of these microtubules. So if we had isolated microtubules and dianin was present, what we'd end up having is a sliding, right? So because it would end up kind of walking along it, so one filament would walk in one direction, the other one would be pushed in the opposite direction. So in order to actually get this beating or power stroke to happen, we have to have linking proteins present. So the linking proteins cause there to be a bend, because as the dianin is trying to walk, you end up getting a bending or flexing of those microtubules. And then as it tries to kind of reset, it bends back. Okay. So it's the dianin with these linking proteins that allow for the cilia and the flagella to have its beating, its power stroke movement. So our last filament to talk about in cytoskeleton is actin. So the way actin is going to assemble is that the actin monomers are going to be added end on end, and this is done in kind of a twisting pairing of this. So we can see here on our panel B, Just like with our microtubule filaments, we have a plus N versus a minus N. And so panel C, you can see what they've done is colored the monomers different colors. So you can start to see how there's a kind of a twisting that occurs so that they're offset from each other. And just like with microtubules, we had to have, a molecule to help to form bonds and to provide energy in order for those bonds to occur, and that was GTP. For actin, it's going to be ATP, and so on the right side, we can see our ribbon model of the actin and how we have our ATP present, and as this becomes a filament, that ATP is going to become ADP. And again, just like with the microtubules, this hydrolysis that happens allows for there to be that polarity, a plus end and a minus end. So, as we mentioned before, actin is going to be found in highest concentration near. So, as we mentioned before, the actin filaments are going to be found towards the outside of the cell near the plasma membrane at the highest concentration. And so, because of that, it's going to act kind of like the walls of the cell, and not the cell wall, but it's going to provide the shape of the cell, and it can be rearranged quite readily so that if the cell has to change its shape or move, actin would play a role in that. So in panel A, we can see the microvilli, and we had talked about how the projections that can be formed on the cell, like microvilli, would actually have actin to create that shape. Panel B, we have the actin filaments coming together to create stress fibers, so that these could contract and allow the cell to shift or flex due to different stresses put on. Most likely at these kind of points where these fibers are coming together, we would also have structures that are present, like anchoring structures between other cells, again, to allow for these stress fibers to have more of an effect. In C, what we're seeing is the pseudopodium or filopodium. So filopodium, filaments, podia, legs. You stand at a podium. And so pseudopodium are kind of like false legs. So again, filopodium and pseudopodium are interchangeable. But you can see how we have these actin filaments that are moving into that area, allowing for that plasma membrane to change its shape and become these projections off. And so the ability to create pseudopodium or filopodium for movement would be really important for like an amoeba, which is a protozoan, but also for our own cells, our immune cells, when they are engulfing, like our immune cells, our white blood cells would have to engulf bacteria or pathogen, and so the ability to wrap around and create these projections that could then engulf the cell would be really important. And so the rearrangement of those actin filaments allows for that to happen for our immune cells. And then when we're looking at cell division, that contractile ring that forms where we get the pinching off of the cytoplasm and the plasma membrane to create those two daughter cells, actin filaments are making that up. So just like we saw with the microfilaments, with the plus N and the minus N, it's going to depend on whether the molecule is in a T form or in a D form. So for actin, because we're looking at ATP, on the plus N, we're going to have our ATP. On our minus N, we're going to have ADP. And so on the plus end, we're going to have a faster on rate and a slow off rate. And so that ends up having growth in that direction, where on the minus end, it's going to have a slower on rate. So ADP just is not going to be able to bind and polymerize as readily as ATP. And then there's a faster off rate, right? So again, that K on versus K off. And so on the minus end, this is where we're going to have either no growth or very slow growth. And as we saw with the microtubules, with actin, we can also have this treadmilling effect that can happen. And so again, this is where we have one monomer adding at the same rate that one monomer is being lost or coming off. And so there's no net gain, but your subunits are moving down in position. So as you can see here, what was the first monomer is moving into basically the second position, the third position, as ones are adding on. And so there's no increase in length, but there's movement potentially of that. So just like there was drugs that would affect microtubules, there are going to be drugs that are going to affect actin filaments. So Phalloidin is probably the most commonly used one, as far as if we're trying to look at the dynamics or the role of actin in a cell, so in the lab. So you would treat the cells with Phalloidin, and what it's going to do is bind and then stabilize those filaments so we can't get rearrangement. So the cell, if it need to make pseudopodium, won't be able to make pseudopodium. Okay, so there's also a number of proteins that can bind actin that will regulate how actin behaves. So keep in mind that we have monomers that are going to come together to form the filaments. So there are going to be monomer sequestering proteins. So obviously, if we're able to get more monomers together, we can make more actin filaments. There's also going to be nucleating proteins. So these are going to cap and stop progression of the actin filaments for monomers being added in. We can also have severing proteins. So these are going to break the actin, so we'll end up with small actin filaments. We can also get cross-linking proteins, so this allows for there to be like intersections in the highway, right? So if actin's being utilized, you can have a motor protein moving to a different location. Okay, we can also have capping. So capping would be on the positive end. So this would stabilize the actin filament so it couldn't be elongated anymore. So we can also have motor proteins like myosin. They're going to bind to the actin filaments. So this allows for contractions, and we'll look more at myosin and actin related to muscle contractions shortly. And then we can also have these bundling proteins. Bundling proteins are important when we're getting those pseudopodium or filopodium being formed, because we need to have a structure that's going to be able to push the plasma membrane out. So again, as I mentioned, actin capping proteins, they're going to cap that positive side so that we can't get increased polymers being added onto there, so we wouldn't get growth. This is just showing you the pseudopodium that is being created by the actin. So as actin is being added in the direction which the pseudopodium is coming out of. For the case of this figure towards the right, we have our pseudopodium. So this cell is going to be moving from a left to right direction. At the same time, we're going to be having contraction because we're going to be taking actin monomers off of the filaments on the left side, so that the organism is then creeping along, okay, from a left to right. And the actin filaments are being reused, if you will. So we need actin to move in the forward direction, so we're going to take actin from the rear and move it. And so again, here we can see how we have our pseudopodium being formed. So another protein that's important when we're talking about actin filaments are ARPs. These are actin-related proteins. So they're important because they're going to allow for branching to occur, so that we're not just getting like filaments in straight linear direction. ARP complexes, as you can see in panel B, will bind on to the actin filaments, and this will then initiate additional actin monomers to bind to it to then form a new filament on there, okay, so that we can get this branching pattern that you can see. So the branching pattern that occurs because of the ARPs is at a 70-degree angle off of the original actin filament that it originates from. Okay, so it's just because of the conformation of the ARPs, or the proteins. So when the actin monomers bind to the ARPs, and then they're adding on along with that twist that occurs between the monomers, again, the angle that's created is a 70-degree angle. So there is what's known as a nucleation between the actin filaments from other actin filaments. So it's not that there's direct contact. It's the ARP that is allowing for the interaction between that original actin filament and the newly formed actin filament to create these what will end up becoming these actin sheets. So sometimes in the pseudopodium, you can see these ARPs playing a role in the formation of the pseudopodium. So if you had an organism that was defective in these ARPs, they wouldn't be able to make pseudopodiums and make these actin sheets like other organisms could. So for actin, the most common motor protein is going to be myosin. So myosin, similar to dianin and kinesin, are going to walk towards the positive end. So it's like kinesin, where it's going to walk vesicles towards that positive end. So the positive end, if it's anchored towards the plasma membrane, as we see in panel B, myosin, its head domain will be attached to the actin, and the tail domain attached to the vesicle, and it will move towards there. At the same time, the tail end could be anchored towards the plasma membrane, so that this would allow for a movement of the plasma membrane, contraction and shifting of the cell. So the ability for myosin to move is going to be powered by ATP. So ATP is going to get hydrolyzed to ADP, and that's going to give the stroke, allow for that movement of that filament by the myosin. So the exact the details, there's more details in here. If you want to talk about them, we can, but there's a lot of other information. So I don't plan on adding that onto the exam. If you were taking A&P and you're talking about muscle contractions, sometimes, you put in about the power stroke and question or two about that. I don't foresee that being on the exam. So that's going to conclude the lecture for the background information for Chapter 17 on the cytoskeleton. I will be doing another lecture that's kind of looking at applying this into a biological system, and it will have components of cell signaling, which is Chapter 16, and also muscle contraction, so actin filaments in work. But the lecture information in this audio lecture is really the background information, and I hope everybody has an understanding of the information in the slides that have been presented. The other audio lecture is kind of taking that to another level. So if you're listening to the other audio lecture and you're like, "Ooh, I'm getting a little bit confused," it's come back to this lecture, understand the microfilaments, understand the actin filaments, and the microtubules. Okay. Thank you very much.